Hi everyone, today we're talking about GFlowNets for biological sequence design, and Sebastian really has prepared this material very nicely for us. So I hope you enjoy it as much as I did.
Okay, yeah, thank you very much. Thanks for giving me the opportunity to talk about this. And yeah, please, so I'm not sure if I can see if you raise the hands, so you can just interrupt me if anything's wrong or if you have a question. Because sometimes I don't see that or I don't hear that. So yeah, but feel free to interrupt at any time. And yeah, please also feel free to correct. It's very much work in progress for me. I'm still learning. So yeah, but I hope we have a good experience. Okay, I'm going to share my screen then.
Yeah, well, to make sure that everything's correct, we are very lucky to have the first author of one of the papers here, Max Schein.
Yes.
But then, before you get started, do you maybe quickly want to say who you are? You're at MPI.
Yeah, sure.
Neuroscience.
Yes. Can everybody see the slides already, or should I turn them off? Sorry.
Well, we can see the slides.
Okay. Yeah. Cool. Okay. So I'm Sebastian. I'm currently working as an AI research engineer at MPI for biological cybernetics in the systems neuroscience group, and I am trying to think about large-scale neural dynamics in neural networks, but I also want to get acquainted with methodology from the drug discovery field and all sorts of AI, because I think there's great potential everywhere in the field to communicate and to understand how methods from one field can influence the other and inspire the other methods. So, and this is actually how I got interested in GFlowNets in the first place. Okay, let's start. So the goal of this presentation is to give you an outline on GFlowNets. It's of course not a thorough discussion of all the mathematics behind it, but the goal is to equip you with an intuitive understanding and visual understanding of what goes on and how the mathematics behind GFlowNets work in order to equip you with kind of a methodology to think about and maybe design your own experiments. So I'm first going to try to talk about the goal the intuition behind GFlowNets very short. And then the largest part will be the fundamental ideas and the reasoning behind the big GFlowNet Foundations paper by Benjio et al. And then I'm going to try to translate these fundamental ideas into how to design a practical study with the example of this biological sequence design by Jane et al. The author is present also. Thanks a lot. And then I'm going to give you a little personal perspective on why I'm interested in GFlowNets and how I think they could also be applied potentially in neurosciences, and a quick summary. And if time still permits, but probably not, some extensions to the original paper because I can't cover everything. It's a very, very lengthy paper. So the original idea is basically that you want to sample a composite object denoted as X sequentially by learning a distribution over the object space that matches a given reward function. And the idea is basically that at the end of the process, when the GFlowNet, which is a network that parameterizes flows from which you can build these distributions, will match the reward functions in the end. And you sample this object, which is composite, sequentially by piecing together basically objects that are not fully constructed yet. And of course, to do this, you need a sampler, which is a generative policy pi a, which depends on the state S. And the state S, and I think this terminology is borrowed from reinforcement learning, the state S now means a partially constructed object. And an action is basically the choice which piece of the next object to attach. And it's to the partial object. And this terminology is very general. So if you think about objects, you can think of a very general class of things you might want to think about. It can be trajectories in a state space. It can be molecular graphs. It can be all sorts of objects that you might want to be interested in. And the framework is so general that it allows for all kinds of different things that you might want to apply this to. And the condition under which this works is, of course, that the reward function allows for generalizing to discover unseen modes. If it's completely unstructured over the object space, then GFlowNets won't benefit from parameterizing a function that may be able to generalize. And other competing methods such as MCMC will work maybe just as well, but if it can benefit from function approximation and thus generalizability, it might be useful to use this approach. So now, what are the applications? The basic application, I think this is the context in which it was conceived, is to learn a generative sampler for biological sequence design. But in the big paper, they cover many more applications, such as learning posteriors over the trajectory space or over the object space, and learn energy-based models in conjunction with the GFlowNets, which allow you to compute information theoretic quantities such as entropies or mutual information. And it's also a way to do very efficient credit assignment, because you don't have the problem, well, you sometimes want to have the benefit to learn, to be able to sample from a distribution and thus implicitly learn the distribution itself and not average over expected values from the distribution. So in credit assignment, how this exactly works will become clear a little later. So now let's walk through the fundamental ideas and the reasoning. How do you imagine flow? So what are the stage for GFlowNets? So I drew this little graph thing, which is a directed acyclic graph, which is pointed. And pointedness is a very important property. And as you can see that the graph has to be directed, because if each state signifies a partially constructed object, in the beginning or in the basic framework, you don't want to allow for actions to deconstruct an object again, because then you would run in infinite cycles potentially, unless you interrupt the process, and you would run into the danger of never finishing the actual construction process. And you can also see that the edges from the penultimate node to the ultimate node are shown in these dotted arrows instead of the normal arrows. And there's a specific reason, because these arrows here will come to mean or will become equivalent to the reward function, but this is also going to become clear later. And keep in mind that a pointed directed acyclic graph is a graph that has only a single source node and a single final node. And this is very important. And a trajectory over this graph could, for instance, be the trajectory that walks through node 0, 1, 3, and then up to the final node. And each trajectory you can basically imagine this as a constructive process where in a couple of steps you construct the final object, and you then go to the final node, to the final sync node, where the process ends. And the important property is that each of these trajectories start in S0 and will end in SF. And then you can define an abstract quantity, which is called a flow over the trajectory space. And that is simply a non-negative number that respects certain conservation properties within the graph. And the property that you're looking for is that within the graph, the total flow across all nodes and edges is conserved. And the idea is now that from this measure that you can define over the trajectory space, you can also derive quantities such as the flow over a single node and the flow across the edges, where the flow through a node, you can imagine this as a flow of some physical quantity, such as particles or water. I think this is the analogy that Benjiu in a post on GFlowNets used. You can imagine that the total flow that flows through S1, through each node, is simply the sum over all flows through all trajectories through that node. And the flow along the edge is simply the sum over the flow of all trajectories through that edge. And the important property is now that as long as the flow within the graph is conserved, that means you don't lose any particles, there are no sinks and sources within the graph, to put on my physics hat a little bit, then you can see that the flow through the first node, through the source node, has to be equal to the flow through the sink node. And this also in turn has to be equal through the flow to the flow through all of the trajectories. And in analogy to statistical physics, you can now define a partition function over the space of trajectories. And that means that you simply can count all trajectories, assign the flow, and then construct a probability distribution over the space by dividing through the partition sum, such as you would do in, for instance, equilibrium thermodynamics, when you have a Boltzmann distribution over the space of particles, and then you divide by the partition sum of that space. And
okay.
yeah,
so,
sorry.
can I maybe do my quick, my quick understanding here?
Sure.
So I have my source node. I throw one into it, for example, or like I throw one into it, and then we split up one into wherever a bit of the one goes, and then we maybe have a half in S1 and a half in S2, and it splits up again however it wants, and at the end we have one again. And now this one is just Z, and you're calling Z the partition function. Or well, maybe Z can also be something, some other number that we split up and combine again.
Exactly. Yeah, you will also see I made some example with actual numbers, so you can see this is very abstract right now, but it's hopefully going to become clear later. Yeah, but that's correct. Yes.
Yeah, I have a quick question as well about the analogy to statistical thermodynamics. So typically it would be, partition functions or ensembles and trajectories need to be microscopically reversible, but in this case you have directed graphs, so they're not. Can you comment on that, please?
Well, they are reversible in the sense, so this flow is a very abstract quantity. The statistical physics, I drew the analogy because there is a partition function and there is a measure over the space of partition functions. Of course, the flow in this case is not reversible because it's a directed graph, but you still have a sort of continuity equation that I'm going to go into later that holds between certain parts of the graph. So in that sense, you have a system where the total flow across the node is conserved, but the flow in that sense that it flows in one direction is not reversible. So the analogy is not perfect. Yes, that's right.
Okay, thank you.
Yeah.
Sorry, maybe I can add something more to that. So in addition to like just the sort of standard flows that are discussed in the foundations paper, we do have the notion of like reversibility. We define this thing called a backward policy in like the detailed balance equation, which I guess maybe Sebastian will cover at some point. Yeah. But we do have the notion of reversibility on this graph. Like you can the directedness of the graph comes from the way you want to construct your object of interest, but you can also define like a backward policy, which will be like a distribution. Like at each state, you will have a distribution over your parents, and you just sort of go back in the graph. But of course, that doesn't have any physical notion attached to it, so it's like more of a mathematical construct that we use. But there is that notion of reversibility, though it's not an exact 1:1 correspondence with statistical physics.
Okay, thank you. So if you take all the reverse arrows, you get the reverse distribution. Is that your sort of analogy there?
Yeah, you can think of it as like the ways in which you can decompose the object. So if at the end you have like a complete object, like a complete molecule graph, you can have like backward trajectories, which would be like how you can sort of decompose the object. It's useful for like applications in like training generative like EBMs, energy-based models and stuff. But yeah, I mean, I'll let Sebastian get to that.
Okay, thanks.
Yeah, but very good question. So I think it's a little dangerous. The analogy, because the backward probabilities are not conserved across the nodes, but the flow is. So it's initially when I was learning about GFlowNets, I also thought about this as probability flows, but technically that's not correct. But let's go further, and then it'll hopefully become more clear. So what you now want to do is you want to assign the flow through the penultimate nodes to the ultimate node, or you want to set it equal to the reward function. And if you know that your space, basically the space of objects that you're interested in is covered by the graph, that means that it has full support, and that the flow is conserved across all internal nodes, and you can define the notion of partition functions. So under these three conditions, you also know that you will always be able to find a well-defined flow on this graph that is exactly equal to the reward probability, to the reward function. Well, you will be able to define a flow where the flow from the penultimate node to the ultimate node or the flow through x3, which is the same, is equal to the reward function. And now let's go to the flow conservation equation or flow matching condition. So the matching condition now says, and this is the very central idea, this is one of the 2 or 3 central ideas behind GFlowNets, is that the flow that comes into any node Sj, which is internal to the graph, is conserved across each node. That means the total flow through the node Sj is equal to the total flow out of the node Sj. And this is also from this notion and from the definition of the forward and backward probabilities, you can construct the detailed balance equation that Mark was alluding to, but that's going to come later. So so the idea is now that you want to find a flow function, which is a non-negative function on the trajectory space that is conserved, such that the flow from the penultimate nodes into the final node is equal to the reward function that you are given. And if that is true, then you know that you can simply construct a probability distribution over trajectories by dividing the reward function through the partition function, or the flow through the first node, which is equal to the probability of sampling a trajectory that ends in the object that you're interested in. And this is how you learn the distribution. So it works by having a pointed graph that is equivalent to the problem that you are trying to solve. That means that every node which you can end up in represents one object that you might be interested in, and flow has to be conserved across the graph. So that's the basic idea. And the quantities that you can derive from that now, and this is how you go back and forth in the graph, are the forward and the backward probabilities. And the forward probability is simply defined by the flow from node Si to Sj divided by the total flow, and that's a well-defined probability distribution. And the backward probability distribution across the graph is simply this flow across the edge divided by the node Sj. And this is how you go back and forth between nodes. And this is, you can sample an object generatively by starting at an empty state. And with the backward probabilities, you can also take an object, and if you have a well-defined flow on your graph, you can deconstruct the objects and get the sequence of steps that was needed to get to the finally constructed object through the backward probabilities. And the last ingredient that we need now is Markovian flows. So far, we have been concerned with probability distributions over trajectories in the space that lead to a fully constructed object X. But when you can decompose that probability distribution into individual forward probability distributions, then the only thing you need to worry about are Markovian flows, which are flows from which you can construct these forward distributions. And it turns out, and is proven in the paper, that for every flow that you can find on the graph, there is an equivalence class whose representative is a Markov flow. That means for every flow that you can find a Markov flow that has exactly the same forward and backward probabilities. And that means you can ignore all flows but the Markov flows. And you only have to care about these quantities along individual edges and pairs of nodes, which reduces your search space dramatically. And that's the gist, basically. So I want to make a little numerical example because this has been very abstract, and I want to show you the relationship, which is sometimes non-intuitive, between flows and transition probabilities. And this is also where the analogy to statistical physics breaks down. So let's imagine you have a flow network like this, like Hannes said in the beginning, where you have some initial flow of, let's say, 6 coming through, and then it splits up, and in the end it will be 6 again. And from this, you can calculate the forward probabilities. And you can already see now that you have that the probabilities from each node to each of the child nodes have to sum up to 1. And the same for the backward probability. But the analogy to statistical physics and to reversibility breaks down because you can see that the probability is not conserved across the nodes, but the flow is. Yeah. So the GFlowNet is now defined as an approximation to a Markovian flow that requires a pointed directed acyclic graph, which has unique source and sink states, a reward function which is given, which can be learned, but from the perspective of the GFlowNet, it's given, and a flow parameterization such that the flow parameterization approximates a Markovian flow on the trajectory space. Are there any questions so far or something that's not clear?
Yes, I have a question.
Yes.
It may be a very basic question, but what's the difference between S and X on your graph? Why sometimes your node are S's and other times they're X's?
Good question. So that's simply notation, because in the papers, you will often see that internal states, which are not fully constructed objects, are denoted as S, and the fully constructed objects are denoted as X. But there's, well, all the equations and properties that hold for the internal nodes S for the partially constructed objects also hold for these objects, with the exception that the reward function in this case is only defined over these objects here and not over the internal states. And this is why there's a notational difference. Yeah, sorry for not mentioning that. Thanks for the question. Yeah.
Thank you.
Yeah.
Also in the chat, so we were asking, can you use R of X to model if the total flow into the node is different to the total flow out of the node?
I think, yes, you can extend. Sorry, yes?
No. I was just thinking about the question.
Oh, okay. Yeah, I think so. If you were to use R, if I understand the question correctly, is the question that if you could define rewards that change the flow across the node, for instance, internal rewards or something, because that is possible, but you would have to, I mean, that's an extension to the basic GFlowNet idea, but that is definitely possible because, I mean, you can still learn flows that respect the conservation equations where you have additional source and sink terms here. So I don't see a problem, but maybe, Moshk, do you want to say something? Maybe that's wrong?
Yeah, I'm not sure I understood the question like as it is typed in the chat, but yeah, if the interpretation is like if we can have additional, basically intermediate rewards, then yes, that is possible. Then there's actually a very recent paper from some of my colleagues at Nila on the exact same thing. So yeah.
Cool, thanks.
All right. Anyway, so to me, the question is also not entirely clear, but we also have another very clear question. Are we learning the DAG or only the flow?
Only the flow.
Good.
Because the DAG is defined by where the flow can go. It's basically defined by the action space and the initial node.
Yeah. Okay. So, well, or maybe a quick recap. We calculate our forward probabilities by we calculate our forward probabilities by taking the total flow, like the S, the F of S0, and we take the flow in a single arrow and divide that by the total flow coming into the parent.
Yes, exactly.
Okay, then let's go.
And the backward probability similarly, you would simply divide by the flow that the edge leads to.
Yeah.
This is why this is 1, for instance. That might be a little confusing in the beginning. I was also a little confused, but yeah, there's only a single node, so it has to be 1, right, on the way back.
I mean, if there's nothing else coming into you, then the stuff that came into you could only come from that arrow. Yeah.
Exactly. Yeah. Okay, so now let's talk about the losses. So you can get loss functions directly from the continuity equations, and all the loss functions that you can construct also have to respect the continuity equation at least implicitly. So the basic the basic loss called the flow matching loss, which I think was the first one that was proposed and that was also used in the very first paper on GFlowNets by Bengio et al. From 2021, is the flow matching loss. So it basically requires it basically enforces the continuity equation on the parameterized flow. And
I just sent something to the data.
Okay, I think that's someone who forgot to mute them. Yeah, let's just continue.
okay, yeah. So you basically want to minimize the difference between the inflow into each node and the outflow of each node, which you can see here. In practice, you will want to take a log because sometimes a log of these quantities, and because sometimes that makes the convergence properties nicer, because in the end you will have many more nodes than in the beginning, but this is the basic idea. And what you can construct directly from the definition of the forward and the backward probability distributions and the flows is the detailed balance equation that was already alluded to. So the balance equation basically says, even though the forward and the backward probabilities across the nodes are not conserved, this quantity is, and that is a direct consequence of the definition of these two quantities and the continuity equation. So it says that, and that's very similar to the detailed balance equation in the context of Markov chains. It says that the total flow multiplied by the probability distribution by the forward probability has to be equal to the flow through the target node times the backward probability. And as a numerical example, you could have a flow of 3, and this is actually coming directly from the numbers that you can see here. So it's simply taken from here. You could have a probability of 2/3 and a flow of 3, and this has to match a flow of 2 and a backward probability of 1. And another loss function, and the last one that I want to introduce is the trajectory balance loss, which was introduced by Malkin et al. From 2022. And that can be derived by looking at the probabilities over whole trajectories. So if you have a Markovian flow, which is the flows you are interested in this case, then you can split up the probability over one trajectory, which is equal to a fully constructed object, into these terms and simply multiply them. And the only thing you still need is a prior, which is the P over S0, which is always 1 by pointedness. And the probability of a trajectory of constructing an object in the forward pass has to, or on the way from S0 to the object, has to be equal to the probability back from the object to S. And so you set those two equal, and you solve, you simply multiply by Z, and then you obtain this loss function here. So that's simply the product of the backward probabilities times F and the product of the forward probabilities times Z. And keep in mind that the partition function also has to be parameterized in many cases because it's intractable for very large state spaces. Okay, so GFlowNets. Are there any questions now?
So, yeah, are you saying that these 3 losses, like, do we right now most of the time in our modern GFlowNets use all of them at the same time, or did they replace the previous ones? Or yeah, okay, so this trajectory balance loss is like the best, and that's what everyone uses now.
It was developed for efficient credit assignment, and I think what I've seen, I think in the paper on the first paper they used this one, but it can be a little. It can exhaust your computational resources quite quickly because you have to, in order to calculate this, you have to sum over all the edges, and if that's thousands of edges, it's going to become intractable quite quickly. So you want to focus on relations between pairs of nodes or full trajectories that you take. And I think the standard loss that I have seen used most often is this one, but I'm not entirely sure.
Okay.
So maybe,
yeah, so I think the trajectory balance loss is by the default choice, but in terms of, I guess, the latest losses, it's probably an extension of trajectory balance, which also applies to like partial trajectories. So the trajectory balance loss is defined on complete trajectory. So you have to go from S0 to X. We have a variant called sub-trajectory balance, which for which you only require partial trajectories. So you can leverage any sub-trajectory within a given trajectory, and you can that learning objective results in like lower bias in the gradient, lower variance in the gradients, and it results in relatively better performance for very long trajectories. But yeah,
Okay,
so
nice.
as Sebastian mentioned, like this is like progressively better objectives, so we don't use multiple of these.
Yeah. Okay, but we also have a question from Gabri.
Could you touch a bit more on how you parameterize these models? Because you're saying, you know, I guess here from the way that you suggest here, you're parameterizing these flows and the partition function. Is that the way one would typically do it?
So I'm going to cover that in the next part, because this was the theory, and now that's a very good question. I was going to talk about GFlowNets in practice and what you actually parameterize and what you actually do in practice. Is that your question?
Yeah, thanks.
Okay, cool. Yeah. Any other questions before we move on? Okay, cool.
A quick one. So the number of paths is growing, no? So with the size of the network, how do you typically deal with the exponential number of paths?
The path number is, I think it's not necessarily growing with the size of the network. I think the path number is growing with the size of the action space. That means which actions you can take from a certain state. But I think typically you would want to take a larger network for larger state spaces.
I think the network might be referring to like the state space graph. Like
I see. The graph. I'm sorry. Yes. Okay. Yeah. Sorry. So one way I've seen in the literature, but I'm not super proficient at that yet. One way I've seen is simply taking the logs of these long trajectories because in the beginning you will typically have only a few options, and if the graph branches out like a tree, which happens often during these generative processes, you will want to account for much smaller flows in the end. So when you have very long trajectories, you will simply take a log of these terms in order to account for the flows getting smaller and smaller. But I think Moksh mentioned something else that you can do, such as sub-trajectory balance.
I think so. At least from what I understood, the question is like if the number of as the number of nodes increases, you will have exponentially more paths within the state space graph. So there are two things. One is if given the conditions of GFlowNets are satisfied, so basically you have support over the distribution, and you train it for long enough, the number of paths shouldn't be a problem because like eventually you will converge to like sampling from the reward distribution. So that's one thing. Now in practice, as Sebastian mentioned, you might have issues like with learning flows because of either having sparse rewards or B having very long trajectories. And in both cases, the sub-trajectory balance loss, in which you can leverage like partial information from partial trajectories, that seems to do the trick. And within, I guess, empirically, Sebastian will cover this, but we can expect it to work even in graphs where the number of terminal nodes is like 10 to the power 20 or something, at least experimentally. That's what we have seen.
Thank you.
Cool. Yeah, thanks a lot. So let's go ahead. I'm not sure how well we are in time, but yeah, you can interrupt me at any point if it's getting too long. So GFlowNets in practice, as was already asked, is when you have a partially constructed object, then you typically will want to parameterize, for instance, the flow across the edges. But there are different choices of parameterizations, and depending on that, different loss functions that you can pick. But a very simple example would be to simply parameterize the policy itself, or the forward probability distribution, or the flow across the edges. So I think in the first paper written on GFlowNets, yeah, they simply parameterize the flow itself, and or the edge flow itself. So I'm going to explain this in a little detail with a very simple example inspired by the first paper. But yeah, this is simply for to give you an idea of how to do this in practice. So we are drawing from fragment-based drug design, and let's assume you have a vocabulary of two predefined pieces, and you want to build a molecule from that. And your action space, which determines the size of your, in this case, almost tree, is you either can pick piece 1 or pick piece 2. And what you are given is a policy pi and a transition function. And the policy simply picks the pieces, and the transition function is a given function that basically tells you or tells the network how to construct the next state. But what you want to parameterize is the flow. That means the generative policy, and typically not the transition function. And a state space for this could look like this. So you have an initial node, and then you construct the objects along these paths. And I said it's almost a tree. Sometimes you will read the state space looks like a tree, but that's technically not 100% correct, so don't get confused. It is almost a tree, and it looks like this because all the flows have to end up in the final node. So it's still a pointed graph. And yet what you will do is pick an action from the policy and then construct the next object and do that in the next iteration as well and get a reward for the final constructed object. And let's say you take this path here as an example, then you would have the flow that is parameterized, that is simply a neural network in most cases, that goes into the node, which is simply the flow along this single edge here. And the outflow of this, if you look for the trajectory balance loss, then it will be simply the sum over the flow along these two edges here. And the first loss in your trajectory loss, constructed from the continuity equation directly, is simply the difference between these two terms. And in the next iteration, you will have a flow along this trajectory that you want to be equal to the outflow of this node into the final node, which you want to be equal to the reward. That means the second term in your loss, in this simple example, will simply be the difference between the reward and the edge that goes into this final node, the penultimate node. And your total loss will then simply be the sum over these two partial losses along the trajectory shown in red. And this is how you do it in practice, really. So in this case, at least, but there's different possibilities. But in this case, at least, you will parameterize the flow itself.
Okay.
Yeah. Does it clarify?
So here we only parameterize the flow.
Yeah.
Is that correct? So
yeah,
we don't parameterize the partition function. Okay.
Exactly. Yeah.
But then Moksh has a point as well, I think.
Yeah.
Yeah, so sorry, could you go back to the previous slide? I just wanted to clarify one thing. So the final node that you have in the DAG, it's not, or the tree in this case, it's not necessary. So all of the sort of math works out equally well if you just assume that all the terminal, all your like fully constructed objects are terminal nodes. So you have like a lot of terminal nodes basically. And instead of having the condition be that the outgoing flow from the fully constructed objects is sort of equal to the reward, the condition just becomes the incoming flow into the sort of terminating states is the reward. So that final node is not necessary, which is why like you will see the mentions of the state space being a tree, because it is actually a tree. You don't need that final node. Like everything works out even if you don't have that final node.
Okay, yeah, thanks for clarifying. Yeah. Okay, any more questions at this point? Okay, good, then let's go on. So now let's go to the biological sequence design by Jane et al. So the goal is to propose a diverse set of candidate sequences that achieve high scores on a given external goodness function. And why is diversity important? As you already mentioned, you want to catch the modes of the goodness function. And often the success of these candidate drugs that you're sending through consecutive trials after the candidate proposal in silico is that they have to satisfy multiple desiderata that can be very difficult to model and that are sometimes even conflicting. And so what you want to aim for, if you don't have a model of the biological processes and the interactions that go on during these post in silico design trial phases, you will want to propose a maximally diverse set of candidates that achieve high scores in the given goodness function that you are interested in. For instance, high fluorescence intensity. That was one of the examples in the paper. And the diversity is, it can be achieved by using GFlowNets because they are able to learn more of the nodes and also do that quicker than other techniques that are also discussed in the paper and that are direct competitors. And the setting now is that you have limited label data, and you also want to combine uncertainty estimation in order to encourage exploration with the sequence generation to catch the high goodness function value modes. And from that, you're going to get a reward function proxy and try to learn these design sequence tasks, these biological sequence design tasks. So the setup looks like this. It's a little complicated in the beginning, but yeah. So you have a labeled data set in the beginning that consists of the ground truth sequences and the labels y, which is a ground truth score that is given by the goodness function. You also have a usefulness measure, this goodness function that is called oracle in the paper, that can take in these molecular graphs and output a value for the goodness function. And sometimes the oracle is learned and sometimes it's externally given, but it's not part of the GFlow learning process itself. Then you have a model that that gives you the reward proxy, which is used to train the GFlowNet. And in the paper, they use different combinations of techniques, such as Monte Carlo dropout and deep ensembles, to learn a model that does not only give you an expected value of the oracle, of the goodness function, but also an estimate of the uncertainty associated with that value. And an acquisition function then constructs a reward function proxy from these two functions. And it can be as simple as upper confidence bound, which simply adds mu to a scaled version of sigma of the uncertainty. And the higher the scaling value is, the higher the reward will reward uncertainty and thus encourage exploration. And then you also need a parameterized GFlowNet. And you need predefined desiderata, one of which is the usefulness measure, but the other ones are diversity and novelty, at least in this paper. And diversity intuitively simply means how far away are all the candidates that have been proposed from each other in the space of candidates. And novelty means how far away are they collectively from the ground truth set.
So
yeah,
now this is something that we can build into our reward. No.
So you build the reward proxy from your estimate of the goodness function and the associated uncertainty of that estimate. Because you want the reward function, at least in this context, not only to capture high goodness, which is one of high usefulness, for instance, high fluorescence intensity, but you also want to reward, in parentheses, in quote unquote, high uncertainty, because that will encourage exploration. Yes, in the space of candidates, lead to more diversity. Yeah.
Yeah, but so, now the diversity and novelty is already built into your oracle or not? Or is it just something that you build into the uncertainty, so to say, of your proxy?
So as far as I understood, it's not built into this process here, only implicitly maybe through this. But what is given is the usefulness measure. And what the GFlowNet is supposed to learn via this procedure is to produce candidates that automatically, without being told to do so, produce candidates that are high in diversity and novelty scores. But you don't train with these scores. Is that correct, or did I misunderstand?
Yeah, that's right. So just to maybe add some additional context, the oracle is assumed to be like expensive to query. So like you can think of it as like actual wet lab experiments, which can take months to run, or you can have like computational, like expensive computational simulations. But the point is like it is super expensive to compute, and which is why we cannot directly use it as the reward function for GFlowNet, because for training GFlowNet, you require like fairly large number of samples, and it's not feasible to train directly on the oracle, which is why we introduced the surrogate proxy. As Sebastian correctly mentioned, the diversity and novelty are essentially just what we expect at the end. So they are like evaluation metrics, not necessarily things that the GFlowNet optimizes for specifically. But those are the things that we want from such a system.
But a strength of GFlowNets is still that we can have this arbitrary reward, or well, in this case, maybe not reward. We can have this arbitrary oracle with which we define our reward, where we can use all sorts of metrics that we want, and we're entirely unrestricted.
Yes, so but I guess the setting in which this operates is like we are generating a batch, and
sure.
like so the reward would be defined like the diversity you can only define based on a batch. So if you were to generate a batch with the GFlowNet, then definitely you could like have like components of diversity within your reward function to like explicitly encourage diverse generation. But I guess the point we tried to make in the paper is that you don't necessarily need that. Like just sampling from the reward distribution allows you to sort of benefit from this diversity as well directly.
And then Karen is asking, do we have any guarantees of discovering the modes of the oracle?
So I guess like we don't have any like exact guarantees, but if we assume that the policy has support over like the reward distribution, then and if your neural net has enough capacity, then we are guaranteed to converge to like the target distribution. That's all the guarantees there are. So like that is basically like the main GFlowNet theorem. But apart from that, there is no specific guarantee on, like, let's say, for example, the number of samples you require to match the reward distribution and so on.
Okay.
Yeah, cool. Yeah, thanks a lot for clarifying. So what you want to do in practice is you want to fit a model to the data set. So this is there's two loops to this algorithm behind that. There's an inner loop where you train the GFlowNet with the methodology akin to what I explained in the practical example with the tree, and you have an outer loop where you train the actual model on the data set, because you can't query the oracle an infinite amount of times. So you want to get a model proxy and train the model on the data set with the labeled ground truth data, and then you train on the reward proxy, you will train your GFlowNet, and then you will sample a batch, not exactly, I mean, you will sample a batch from the GFlowNet, you will generate parts of the batch, and you will also sample parts of the batch from the ground truth data, have it evaluated by the oracle, which is either given or it is pre-trained on the data set, so you can query it, and then you will add that to the data set. And what happens when you train the GFlowNet and sample these batches that you will send through the oracle for evaluation and add it to your data set is simply that you, with a certain given proportion, which changes over training, you will sample trajectories from the policy directly with a little exploration encouragement here, which is inspired by the idea of like epsilon greedy, as far as I understand, in epsilon greedy exploration in RL. And for the other fraction, you want to sample trajectories from the data set itself. And therefore, in order to be able to do this, you have to parameterize the backward policy as well, because if you sample from the data set and you want to reconstruct the sequences, then you need the backward probability as well. And then you combine both sets, compute reward, and update the parameters according, in some subset of experiments, to the flow matching loss, and in the other experiments on the trajectory balance loss. And the cool part about this is that it switches from an active to a non-active learning schedule, and from an online scenario to an offline scenario, by having this parameter gamma, which increases over training. That means in the beginning, you have small gamma, and that means most of your trajectories are sampled from the policy itself. It's basically an on-policy active learning setting, and that goes over training into a more offline setting where you sample a higher fraction of the trajectories that you add to the data set from the original data set itself. And your network, the reward that you will catch, the reward that you will get in the end depends on the value of gamma that you choose. And is there any, I have a question on this actually, is there any specific reason why can you explain why gamma equals 0.5 turned out to be the best? Is there any intuition behind that, or is it just a result that that has no explanation yet?
Yeah, there's two clarifications. So one is the gamma does not change over training steps, at least in this paper. It like the gamma is fixed during let's say like the training of the GFlowNet. So we have like fixed proportion of offline samples that we take in the batch. That's one thing. Now, so the reason for that is like the gamma, like within so this entire training of the GFlowNet is like one step within the active learning loop. So we train the GFlowNet for one step, we generate a batch, we sort of query it with the Oracle, we update our data set, then we train the GFlowNet again with this updated reward function because we have observed new data. So we have this sort of outer loop which you mentioned. So within a single sort of GFlowNet training, you don't necessarily benefit from changing gamma, but of course that is something which could be interesting to experiment with, but we did not do that in this paper at least. So here the gamma is fixed over the training steps of the GFlowNet. And I guess the reason for not having or like one of the, I guess, working hypothesis for why like 0.5 seems to work the best is so there is always this delicate balance of exploration and exploitation, like in RL as well. And the problem is if you just train on most of offline data, basically, your policy will be sort of constrained to just sort of be in that region, and you might suffer in exploration. Of course, like if we train this for long enough with like large enough neural networks, you would expect all of them to converge to the same thing. But considering the fact that, like, I guess due to practical constraints, you can only train for finite number of steps and with finite number of trajectories. So your trajectories are not necessarily representative of the entire distribution of sequences. That is why we sort of don't gain that much. So this leads me to the second point. So like I mentioned earlier, one of the key criteria for like GFlowNets learning the right thing is you need to have support over your distribution of trajectories. So this feeds into the, I guess, the second line where we have this uniform exploration, epsilon uniform exploration. What this does is it basically ensures that this forward policy PF has full support over the distribution of trajectories. So basically, eventually we'll cover all possible trajectories, like after like enough training steps. And that also partially explains why like gamma equals 0.5 is sort of working the best, because if the sort of offline trajectories that we have in our data set had like full support over the distribution of trajectories, then we would expect it to like learn be basically same as the on policy reward. But since like we have like this biased data set, we don't gain much from it. Hope that explains the weird 0.5
yes,
phenomenon.
Thanks. So one question back for that. So I understood that gamma does not change in the inner loop, but it does change in the outer loop. Is that correct, or does it not change at all? Did I?
Oh no, in the experiments in the paper, it does not. But
not at all. Okay,
I mean,
I see.
Yeah, it's fixed for like one entire like experiment.
Okay, I see. Thanks. Okay, I think I'm running a little bit out of time. So do you want still to hear an overview over the experiments, or should we go over to Q&A now? What do you think?
I would be very interested in an overview of the experiments, but I think I also am missing a very crucial point of understanding right now as well. So why do we do this with the data set and the sample trajectories? Why don't I just sample trajectories and let my reward or my learned reward function, right? That's super quick to evaluate them and then use those as rewards to train. And instead I have this data set.
Maybe I can answer that. So the reason for that
also learn proxy. So sorry. Yeah, please go ahead.
Yeah, sorry, I was just saying that we want to train it faster. That's the only thing. So you're like, if you just train it on policy, it might take a bit longer. But if you use offline data, which is basically already trajectories which you know are good, like you can sort of filter them out as well. It can result to like faster training in regions that you care about. That's all.
Okay. Fair. Then let's move to some results.
I'll make this quick. So there are 3 tasks that they looked at in the paper, and the first one is the antimicrobial peptide design. That's small proteins. So you have a fixed length of 50 and a vocabularies of 20, which are the proteinogenic amino acids. And the usefulness measure is directly taken from the database of antimicrobial and cytotoxic activity paper here, the database. And the oracle, that is trained on the baseline data set, is a pre-trained transformer taken from the literature. And here you can see results. So I'm trying to rush through this a little bit because I know I'm running out of time.
Well, why do you have to leave?
No, I thought this was for an hour always.
No. We have as much time as you want.
Okay, cool. Yeah, okay, cool. Yeah, and you can see that against competitors, the GFlowNet in the active learning setting has the...
But wait a second. I do not understand what's going on already. So we're trying to generate antimicrobial peptides. That's our task. Nice. And we have a usefulness measure that works by taking the generated sequence and looking if it's already in this database, or how does it work?
As far as I understand, you have a database with the ground truth, which is the antimicrobial or cytotoxic activity. And in order to predict for your generated samples that are not already in the database, you are trying to learn an approximation to the oracle, which is trained on the baseline data set and pre-trained transformer taken from the literature. So in this case, if you go back to this picture here, you don't only learn the model M that gives you the proxy and the GFlowNet, but you also learn the oracle by training it on the ground truth data, the baseline data set.
Yeah, so we just train our in the beginning we train our reward function on this data set that you have of antimicrobial activity.
The oracle, not
Okay.
Yeah, so the oracle gives you the goodness value, and the reward function is expected goodness plus uncertainty.
Okay, that's good. And trained on baseline dataset and pre-trained transformer.
Yeah,
it's basically okay. So it's just a transformer, a protein language model that takes the sequence as input and spits you out a single number.
Yeah.
Good. Okay, nice. Then we see the results where you wanted to say what?
Yeah, I wanted to say that here you can see that compared to other baselines taken from the literature, I mean, re-implemented, but taken from the literature, is that even though GFlowNet hasn't been explicitly trained to go high in diversity and novelty, it achieves that with pretty impressive results, I have to say. And what's also really cool is that the overall density of the amino acids in the generated antimicrobial peptides is very similar to the known biological densities. So yeah, I think that's a really cool thing that GFlowNet can not only succeed in going high in achieving high values in performance, diversity, and novelty, but also generate biological plausible candidates as far as the density of the amino acids is concerned.
Okay.
And yeah.
Then maybe I think here Gabriele has a question.
Yeah, I just wanted to ask, is the reason why you use this transformer as the oracle rather than the proxy something just to do because it was kind of because you didn't really couldn't get some experimental experiments, or yeah, what was the reasoning? Because this is more
okay. Okay, yeah.
Yeah, sorry. I have two clarifications to make here. One is the oracle is not really the pre-trained transformer from this paper. It's just like a transformer, like just a normal transformer which is trained on DBSP. To provide more context on this task, so the goal here is we want to discover like antimicrobial peptides. But the problem is like there are only like I think 6,000 or so known antimicrobial peptides which have been tested in a laboratory, which are like part of this DBSP database. And the sort of challenge for us was like, how do we design this oracle? So what we sort of ended up deciding is we would have we'll sort of reserve half of the data set, like selected based on target species and like few other biological criteria. And we sort of use that to train like one oracle model, and we sort of use the rest of the data, like half of the data, to use as like a starting point for the GFlowNet active learning sort of thing. So this first half of the data is not used anywhere, like it's just used to train this model, and this model is fixed. So this is like the oracle. The reason for this is like basically just for like making things reasonable in simulation. So like we can actually do this active learning for several rounds if we have a fixed oracle. Does that make sense?
So basically the idea is here you're kind of simulating a oracle, like a more complex oracle, by keeping some data kind of separate from kind of everybody else's. Thanks.
Cool. Yeah. Thank you. Thanks a lot. So any more questions or next task? Okay, next task. So that was the next task was to generate short DNA sequences that have high binding energies with human transcription factors, and they all have length 8 and vocabulary size 4 in this case. So it's a quite small setup. And in this case, the oracle is empirical and exact, since the data set has all the length 8 sequences with the vocabulary size 4 in them. And if you train and compare to other models from the literature, you can also see that with the exception of diversity, GFlowNet in the active learning setting also gets very high. I mean, even in diversity, it's quite high, but not impressively high against the other ones. But in performance and novelty, it's very high. And the model that scores the highest in diversity gets very low novelty scores, very low compared to the GFlowNet. But I also actually have a question to Moksh in this context. Why do you think the diversity here is the GFlowNet doesn't succeed very much in diversity as opposed to the other models? Why it's really good in terms of performance and novelty? I didn't understand that.
Hello. Yeah, so the diversity is mainly in this particular task. So this is like just DNA sequences of length 8. So it's not a very large state space in this example in particular. And like we chose this because people in literature use this as a benchmark. But the main reason is because these are like very small DNA sequences. And like each character can have, each character in the sequence can have only like four possible values that it can take. And the reward function itself, it's basically it's like how well this DNA sequence can bind to the human proteome. Okay, I don't really understand the exact biology of this data set, to be honest. But my understanding is like there's a very few of these sort of DNA sequences which actually bind really well. So there is these two factors which affect the diversity in this particular case. So it's not super expressive in that sense. Maybe that's the right word.
Okay, thanks. Yeah, that clarifies it. And
also,
the last one, sorry.
In all of these experiments, right, when we evaluate the performance in the end, we use our oracle. Is that correct? Yeah.
Yes.
So we don't really like it's not, yeah, we rely on the fact that our oracle, or if our oracle doesn't generalize, so to say, or yeah, if it's, yeah, it completely doesn't generalize, and all the sequences that are out of the distribution of our oracle, those get very low reward, even though in reality maybe they are very good, then
Yeah.
we do not capture this here in the performance.
Yes, so that was the challenge, which is why we had to, like, in the previous task we had to like do the splits ourselves and like sort of come up with a oracle ourselves. And in this particular task, actually, we have an oracle, like an exact oracle, because there is only like 65,000 sequences, such sequences in total, and like people in the past have like actually docked them. So here we have the exact oracle. So like it is the ground truth function. So there is no learned function here which we are evaluating on. So we don't need the oracle to generalize. And I guess the general setting that we envision this sort of GFlowNet active learning loop to be applied in is with like actual real world experiments where, or I guess like some very high fidelity simulations where you expect this oracle to be like the final thing that you care about. Yeah. So I mean, experimental measurements, you expect them to be reliable regardless of whatever sequence you plotted.
Mm-hmm. And like that's because here you just have like, I guess, 4 to the power 8 different options.
Yeah.
And all of them have been already evaluated.
Yes.
Okay. Good.
Cool, thanks. So, and the last task is again a protein design task, which has a much larger state space. The task is to generate high fluorescence intensity proteins, something I've been very acquainted with in the lab over the past 3 years. And the vocabulary size again is 20 and the length is 237. And the usefulness measure here is simply the fluorescence intensity. And the oracle is a model that is taken from the literature. And also here you can see that the performance, diversity, and novelty scores are highest for GFlowNet in the active learning setting. So I would quickly go to the very last point, which is a personal perspective, and then maybe to the summary and the discussion. Is that fine with everyone, or are there any more questions?
Sounds good to me.
Cool. Yeah. So personal perspective is I became very interested in GFlowNets because I'm interested in models of neural internal state dynamics, and there's lots of literature from the machine learning field, but none of them from the GFlowNet perspective yet. And I think applying principles from thermodynamics and statistical physics, especially continuity equations, to the questions of neural internal state and conservation of information in the brain could be very interesting alternatives to hidden Markov models and linear dynamical system models and stuff like this, or the thing that is used right now. And I think one of the main challenges, of course, the length and of the sequences and the exponential sizes of the state spaces. But I think one way to do very efficient credit assignment in this context could be using GFlowNets or GFlowNet inspired approaches coming from the perspective of conservation laws in equilibrium or even non-equilibrium thermodynamics. And the other thing that I would find very interesting is to learn energy-based models, maybe based on GFlowNets, because they have nice properties of discovering many modes, to model functional connectivity in the brain, which is something I've been thinking about for quite a while. Yeah, that would be a very interesting application. I'm interested to maybe discuss with you more about that later on, or maybe discuss later in some personal talks. So if you have any thoughts on that, please hit me up. I would be very interested to talk about this with anyone who has ideas. And now to the summary. The GFlowNets are generative policies that learn reward distributions, and they rely on parameterizing these flows that are conserved across all the internal and the fine penoptimal nodes of the graph. And they can be parameterized very flexibly in order to define various loss functions with them, which are either direct consequences of the flow conservation equations or in analogy to the balance equations that you find in Markov models and also in statistical physics. And they are able to generate, which has been empirically shown to generate very diverse samples. And because they allow their flexibility and the generality of their theoretical framework allows for many extensions. For instance, as was mentioned in the beginning already, you can build inference machines with and without latent variables from them. They are great alternatives to some methods in reinforcement learning that suffer from convergence to only a single mode of the reward distribution. And they also allow for training energy-based models where you train an energy function in conjunction with the GFlowNet and learn these two things in advance. And yeah, you can also generalize them to different settings such as sets and over graphs. It's really flexible, fantastic framework. I encourage everyone to read the foundations paper. Really interesting. Yeah. Okay. Thanks a lot for having me and for giving me this opportunity. Yeah. And now hit me with any questions or discussions. Are there any questions or any points somebody want to make?
Oh, sorry, I was muted, I guess. Yeah, first of all, I want to say thank you for making this, for going through these two papers, preparing this material so extremely nicely and presenting it here so very well. And yeah, I think it was very good for me. I think many others also learned a lot from it, and also thanks Mox for also hanging around and clarifying a bunch of stuff and yeah, answering all of our questions to both of you. All right, but then I think also in the chat I saw something along the lines of what do you think is the main advantage of GFlowNets to compare to other generative models?
Oh, so maybe Mox, do you want to answer this, or should I just give my personal perspective? Because I haven't been involved in the development process. I
already put 3 reasons in the chat, but I would love to hear your opinion as well.
Okay, so from my personal perspective, it seems to be a very efficient method to learn reward distributions. And what I find very appealing is that it has been designed. I don't know if that was the explicit idea in the beginning, because I also heard it was kind of inspired by Bellman by Bellman equations from reinforcement learning. But what I took from this, when I put on my physics hat, so to say, I was thinking about this in the context of thermodynamics, and it inspired me, so to say, to think about credit assignment, and not only synaptic credit assignment, which is a very interesting open problem in neuroscience, but also temporal credit assignment for internal state space trajectories in neuroscience on latent manifolds in terms of these continuity equations, which is something I do not only want to try out GFlowNets, but also it inspired me to think about these problems in the context of thermodynamics, which I think also allows for extending the theoretical framework of GFlowNets from this perspective. Because when you break GFlowNets down and when you look at the continuity equation that is at the heart of GFlowNets, then it's a simplified version of a continuity equation that you find in fluid dynamics and physics, which is the stationary Euler equation. And if you simply go back from that and try to build in the intuition from that equation into extensions of GFlowNet, it could be an interesting alternative to looking at internal state space trajectories in real brains. That's my takeaway. It's a very personal perspective from neuroscience guy, but yeah.
I mean, that is a very interesting connection. I guess I can in addition to the things I mentioned on the chat, which is where we think GFlowNets are useful and where they provide advantage over existing probabilistic frameworks is where we have uncertainty in the rewards. So we're not really sure that this is the right reward function. So this is the case in for example, if your reward function is learned and you don't like you can't rely on the reward function as like your final metric of goodness. And B, if the reward function is multimodal. So if your reward function is just unimodal, then you could just do RL or MCMC. You don't necessarily have a problem with those. And C, you have like compositional structure in the object that you're generating. So things like graphs, sets, and so on. So if you don't have that, then there is not much you can expect your neural networks to generalize. So like those are, I guess, the three criteria of cases where GFlowNets would perform better, or you would expect them to perform better. And I guess moving to connections that you made. So actually there is, again, work from my amazing colleagues at Mila on sort of establishing connections of GFlowNets with, or like actual formal connections between GFlowNets and existing inference paradigms. So for instance, like one of the recent results was that GFlowNets are sort of very intimately connected with variational inference, and in particular, like the trajectory balance objective that we saw is so the trajectory balance objective essentially it maximizes the lower bound on a particular variational objective, and the more important result is that the expected gradient of the trajectory balance learning objective is exactly equal to like sort of minimizing the KL divergence between your forward sampling distribution, so the one induced by PF, and your reward distribution. So there is this paper which formalizes this quite nicely, I would say. And coming to I guess differential equations and like connections to neuroscience. So GFlowNets also serve as like a unifying framework for a lot of other generative models, like for example diffusion, which is I guess very popular now these days. So GFlowNets, or I guess diffusion can be seen as like a particular case of GFlowNets in continuous space with like particular choices of forward and backward noising processes. I'll try to put the link in the chat. But yeah, there are lots of these interesting connections. But I guess so far, if we're talking about things that we have established in practice, I would say, yeah, like uncertainty in the reward, like multimodal reward distribution, and like compositional structure in the things that you're trying to generate. Those are probably three main areas where GFlowNets can potentially provide benefits.
In the chat, you sent GFlowNets and variational inference. Is that the paper that you mean that also has the connection between like diffusion models as continuous GFlowNets?
Just adding 1 second.
Okay, good. Sorry about that. Cool. Then maybe one other thing, have you tried, like you say, if we have uncertainty in the reward function, then GFlowNet might fare better in certain evaluation metrics. But have you tried that as well? Like you actually have access to a ground truth oracle, and then you learn a reward function that is suboptimal, and then or you learn an oracle that is suboptimal, and then you do your actual evaluation with the ground truth oracle while using the suboptimal oracle for training?
Yeah, so that's actually, I think, in probably the initial papers where we have like the exact it's in the first paper. So we have like an exact oracle, and we learn like a suboptimal reward function with it, and we show how we can keep improving on it and like sort of maximize the original oracle function. I guess more on like the real world perspective, we also have like ongoing projects with like our collaborators who are like biologists who are actually synthesizing things that we generate with GFlowNets, and we have like this real world validation for it ongoing, but we still are in the process of doing that.
Okay. Yeah, I mean that's I really like what's going on with this. Like also these real world active learning loops that you've now got going on, I guess, with GFlowNets. Yeah, if we have any other questions, and then feel free to maybe reach out to Sebastian or Moksh themselves as well. And maybe we can go for a last questions. Do you see any relations to optimal transport? Like if we have two different distributions and not just one, anything that already pops into your mind?
So GFlowNets solve like a particular version of the Schrödinger bridge problem, which is, I believe, connected to optimal transport. But if I'm being honest, I'm not super well versed in optimal transport to make like a strong statement about that. But I believe.
How do what's it called, GFlowNet solve a particular version of the Schrödinger bridge?
Well, so if you consider your terminal states as so you start with yeah, actually never mind. I don't think I have enough understanding of that problem to make the concrete statement, but I believe it is in this last paper I sent. It should probably discuss that if I remember correctly, but
yeah, I that's why I was interested in this this paper. I guess they must have some sort of explanation why of something like that. Yeah, like if you go from a Gaussian to some arbitrary distribution, then how does this now fit into the framework of GFlowNets, for example? And then if we have a Schrödinger bridge, then we just go from arbitrary distributions to arbitrary distributions and try to learn the path, the most likely path, so to say. Yeah. Anyway, I think we got through all points, and I can confidently say thanks again for preparing all this material. And yeah, it's really been a excellent discussion today, I think. So yeah, that's all from my side. And then thank you, Sebastian. Thank you, Moksh, and have a nice day, everyone. And oh, wait a second before you leave. No one leave. Merry Christmas, Happy New Year. We have no meeting next week. Merry Christmas, everyone, and see you. But cool, that was definitely an excellent discussion. I'm very grateful that Sebastian and Moksh joined us here for this and answered so many questions. And I learned a lot. I hope you did too, as is often the case with these reading group sessions. And yeah, if you want to ask more questions, then Sebastian was very open towards that. Reach out to him. And besides that, see you guys next year. Bye, everyone.